Hello, good evening, students. So we'll start the session now. Let me share my screen. Hmm. Let me introduce myself. Myself, Ankit Kumar Malik, NPTEL PMRF Fellow from IIT BHU, and the course instructor for the Bioengineering and Interface with Biology and Medicine is Professor Sanjeeva Srivastava from IIT Bombay. So we'll start for the session for the week 7. So the topics for the day is amino acids and proteins and proteomics, techniques to study protein and proteome 1, techniques to study protein and proteome 2, and bioinformatics 1. So we'll start from the doubt session with question 1 of the last year assignment. So the question 1 is pick the correct combination of statements that best defines the quaternary structure of a protein. So the statements are: so when two or more polypeptides join forces via disulfide connection, a quaternary complex is created. So second statement is one of the amino acids required for the creation of quaternary structures is cysteine. And the statement third is quaternary structures is formed between peptide linkages where hydrogen bonds play a crucial role in the structure formation. And the statement 4 is aquaporin is not considered as a quaternary structures, but hemoglobin is. So the correct options, the possible options are given here are 1 and 2 statement are correct, first and third correctment are correct, first, second and fourth statement are correct, and the last option, third and fourth statements are correct. Are correct. So anyone would like to answer this question? Anyone?
1 and 2 are correct, sir.
Okay, so let me proceed then. So let me first discuss about the quaternary structure for protein. So this is the quaternary structure of a protein. So basically the quaternary structure of a protein refers to a spatial arrangement and interactions between multiple polypeptides chain. So basically that is subunits. So that come together to form a functional and a biological active protein. Sorry. So not all protein exhibit quaternary structure. It is particularly characteristics of proteins composed of more than one polypeptide chain. So here are some like bonds called salt bridges, hydrogen bond, van der Waals forces, and disulfide bonds. So here are some key points which I'll tell you now to understand about the quaternary structure. So multiple subunits. So here structures are there. Some secondary structure is there, tertiary structure is there, and quaternary structures is there. Basically, we focus on the quaternary one only. So protein with quaternary structure consists of two or more polypeptide chain, each of which is referred to a subunit. So these subunits may be identical like homodimers or maybe homopolymers or different like heterodimers or heteropolymers. So interactions between these subunits is essential for stabilizing the quaternary structure of that particular protein. So these interactions can include hydrogen bond, sulfide, disulfide bonds, ionic bonds, and hydrophobic interactions, as I told you in the last structure. So there are some functional consequences also. So the quaternary structure is crucial for the overall function of many proteins. So it often enhances the efficiency and specificity of the protein's biological activity. And the let me tell you some examples of protein with quaternary structures. So the first example is we call hemoglobin. So hemoglobin is a classical example of a protein with quaternary structure. So hemoglobin is composed of four subunits, each containing a heme group for four oxygen binding. So then the next example for quaternary protein structure is DNA polymerase. So DNA polymerase is an enzyme which involved in DNA replication, consists of multiple subunits working together to catalyze the synthesis of a particular DNA. So now come back to the questions with the statement. So, uh, hmm. So here the statement 1 and 2 according to this, when two or more polypeptide join forces, why does disulfide connection a quaternary complex is created, as I showed you and told in the structure. And the statement 2, which is one of the amino acids required for the creation of quaternary structure, is cysteine. So these two correct combinations for quaternary structures is correct. So rest third and fourth are incorrect about quaternary structure of a protein. So the correct answer for the question 1 is the option A. That is 1 and 2 statements are correct for the quaternary structure of a protein. The correct answer is
Excuse me,
the
sir.
option A. So moving forward to
Sir,
the next
why
question.
fourth is not an answer?
The question second states that which one of the following amino acids lack optical activity. So assume that you are involved in the production of antimicrobial peptides and you wanted to measure the peptide amounts using the UV visible spectrophotometer after purifying them. So which of the following peptides will be the easiest to find at a monochromatic wavelength of 280 nanometer? Choose the appropriate set of responses. So don't confuse students here. So here four structures are given below. Don't confuse with these naming like GLS, BAAKK. So this is the coding for the different structures. So this the first option is A is the coding for this structure, and the second one is the coding for fourth structure, and the third one is the coding for this third structure. Don't get confused with what is this GLSS, YAP, and RLC. There's nothing. This is a coding for some different structures. So we'll focus on the question only. So which of the following peptides will not be the easiest to find at a monochromatic wavelength? So to understand this concept, we'll look into some aromatic amino acids. So to determine which amino acids lack optical activity and which peptide will be the easiest to measure a UV visible spectrophotometer at 280 nanometer. So let's examine the property of some amino acids and peptides. So before that, let me define you what aromatic amino acids are. So an aromatic amino acid is an amino acid that include an aromatic ring. So aromatic amino acids absorb UV visible light and beyond 250 nanometer and will fluorescence under this condition. So this characteristics is used in a quantitative analysis. So notably in determining the concentrations of these amino acids in a particular solution. So most proteins absorb at 280 nanometer due to the presence of this aromatic amino acid residues. So of the aromatic amino acids, tryptophan, please focus on this word tryptophan. Tryptophan has the highest extinction coefficient. So its absorption maximum occur at 280 nanometer. So the absorption maximum of tyrosine occurs at 274 nanometer. So now let's tie this information together. So glycine lacks optical activity as it does not have a chiral center. So as you can see the structure of this L-histidine. So coming back to the question. uh, So this tryptophan and glycine lacks optical activity as it does not have any chiral center. So according to this, only this L-histidine matches with this. Uh, which one? Yeah, this one, the structure one. No, this structure four. Sorry, this four one is L-histidine, and it does not, and it will be the easiest to find at monochromatic wavelength at 280 nanometer. So the correct answer for the question 2 is the option B, that is stating that fourth structure. Okay, so let's move to the next question. So the next question, third question. So in third question, the question is you have purified a synthetic peptide that has 4 negatively and 8 positively charged amino acids at pH 5.6. So you noticed that when the peptides were treated with an alkaline solution, which of pH 8, where 4 positively charged groups were deprotonated. So which is the current net charge of that particular peptide? And what direction will the peptide move in a gel-based solution with an electric field and is introduced at pH 8? So the possible given options available here are: option 1 is a net charge on peptide is 0 and movement of the protein will not happen. And the option B is net charge on peptide is -8 and the movement of the protein will be towards anode side. And the option C is net charge on peptide is -4 and movement of the protein will be towards anode. And the last option, last possible option for this condition is net charge on peptide is +4, movement of the protein will be towards anode. So according to the question, so the peptide has 4 negatively charged amino acids and 8 positively charged amino acids. So at of pH 5.6. So at this pH, negatively charged amino acids are likely deprotonated, that is ionized, and the positively charged amino acids likely protonated, that is not ionized. So the deprotonation at pH 8, so when the peptide is treated with an alkaline solution, which is at pH 8, so which is higher than its original pH. So four positively charged amino groups are deprotonated. So this means that has some of the positively charged amino acids become neutral due to the removal of that protons. So I have done some calculations for the same. So like let's calculate the current net charge of the peptide now. So initially the peptide had a net charge of 8 positively charged, then -4 negatively charged, that is +4. So at pH 5.6, and after deprotonation at pH 8, 4 positively charged groups are neutralized. So the new net charge is now 0 at pH 8. So therefore the current net charge of the peptide at pH 8 is 0. So, and another thing they have mentioned the movement of the peptide in gel-based solution with an electric field at pH 8. So, at pH 8, the peptide has no net charge due to the deprotonation of positively charged groups. Since it has a neutral charge, it will not be strongly influenced by an electric field in a gel-based solution. So in gel electrophoresis, molecules move based on their net charge. So a neutral peptide will not experience significant movement towards the anode or cathode when an electric charge is applied. So the summary of the entire theory is the current net charge of the peptide at pH 8 is 0, and the peptide will show minimal movement in a gel based solution when an electric field is introduced at pH 8. So coming back to the question, and we'll look at the possible options given there. So according to this, the option A. So the option A is the correct answer for the question 3, that states that the net charge on peptide is 0 and movement of the protein will not happen. So the correct answer for the same is option A. So moving to the next question. Just give me 1 minute. Hmm. So the question fourth. So the question fourth states that, so the image below shows the differential in gel electrophoresis that is called DIGE method. So which employs two types of tissue samples, that is normal and the cancerous one. So Psi2, Psi3 and Psi5 cyanine dyes were mixed with the normal cells and the cancerous cell, and the combination of both normal and cancerous cell respectively. So before applying the samples to the gel, they were mixed. So after electrophoresis, gel picture was examined for three distinct wavelengths according to cyanine dyes. So select the right answer. So the given options here are, the option A is the proteins in gel N and C are entirely distinct. So here we are calling N, we are referring N as to normal cells, and we are referring C to cancer cell. Okay. So the option B is the normal of the number of spots in gel N plus C is always smaller than both gels N and C, and the option C is the both N and C gel excludes the probability of batch to batch variation and is considered to be a good reference for further data analysis. And the option fourth is both B and C are correct. So we'll look into some theories what differential in gel electrophoresis is, and we'll come back to the consequences and conclusion for the same. So before moving forward, so let me explain what DIG is. So differential in gel electrophoresis is a form of gel electrophoresis where up to the three different protein samples can be labeled with size matched, charge matched spectrally. So resolvable fluorescent dyes, for example, Psi3, Psi5, Psi2. So prior to two-dimensional gel electrophoresis. So the three samples are mixed and loaded into the isoelectric focusing chromatography. So for first dimension, and the strip is transferred to a SDS page. So after the gel electrophoresis, the gel is scanned with the excitation wavelength of each dye, one after the other. So each sample can be seen separately. If we scan the gel at the excitation wavelength of the side 3 dye, we will see in the gel only the samples that was labeled with that particular dye. So this technique is used to see changes in protein abundance, so post-translational modifications, and any modification that might change the size of isoelectric point of proteins. So the binary shifts might be left or right, or vertical or diagonal. So reciprocal labeling is done to make sure the changes seen are not due to the dye dependent interaction. And the last thing is batch to batch variation, so which refers to differences that may occur between different experimental procedures. So what are the given options here? Here is this one. So I've told you the last statement is batch to batch variation, which refers to differences that may occur between different experimental procedure. So according to this, so in the option C, where both normal and cancerous cell excludes the probability of batch to batch variation and is considered to be a good reference for further data analysis. So the right answer for the question 4 is the option C, that is a batch to batch variation. So moving to the next question, the question 5. So question 5 states that in a mass spectrophotometer, so the ionization source, mass spectrometer, the ionization source performs which of the following functions? So please select the appropriate ionization technique involved with the mentioned activity. Pick the correct answer from the following options. So the correct first option is convert the analyte into gas phase ions in vacuum. This method is called soft ionization. And the statement 2 is convert the analyte into liquid phase ions in vacuum. This is called the hard ionization. And the statement 3 is in MOLDI. Actually, MOLDI here is called matrix assisted laser desorption or ionization. So in MOLDI, the analyte is mixed with aromatic matrix and bombarded with a laser. So this is called soft ionization. And the statement 4th is in MOLDI, the analyte is mixed with aliphatic matrix and bombarded with fast beam of argon or xenon atoms. This is called hard ionization. So any student with some chemistry background would like to answer this question. This is a basic question. So here some definitions are wrong, which is of soft ionization and hard ionization. So any student would like to answer this question with a basic chemistry background. So this is a very easy question. Those who know about some basic principles and working of mass spectrometer. Okay, let me explain. So we'll cover this question with statement to statement. So here the first statement says that converts the analyte into gas phase ions in vacuum. This method is called soft ionization. No, this is not called soft ionization. This is actually called electrospray ionization. Wait, this one. So this is called electrospray ionization. So basically, electrospray. Let me correct this statement also. That why electrospray ionization? Because electrospray ionization is a soft ionization technique where the analyte is converted into gas phase ions in vacuum. Okay. So this is not called soft ionization. So statement 2 is converts the analyte into liquid phase ions in vacuum. This is called hard ionization. So this statement is also not accurate. So ionization methods typically convert analytes into gas phase ions, and the liquid phase ions are not common in mass spectrometry. So this statement is also not correct. Let's come to the statement 3 in MALDI, the analyte is mixed with aromatic matrix and bombarded with of laser. This is called soft ionization. So this statement is correct one. So in MALDI is a soft ionization technique where the analyte is mixed with an aromatic matrix and ionized by laser. So let me explain the MALDI structure and its principle first, then we'll come back to that statement 3 and 4. And statement third is the right one. Just a minute. So, is my screen is visible?
Yes, sir.
Okay. So, here you can see the principle and structure of the MALDI, that is matrix assisted laser desorption and ionization. So, in mass spectrometry, so it is an ionization technique that uses the laser energy absorbing matrix to create ions from large molecules with minimal fragmentation. So it has been applied to the analysis of biomolecules such as biopolymers like DNA, proteins, peptides, and carbohydrates, and various organic molecules like such as polymer, tentrimers, and other macromolecules. So which tends to be fragile and fragment when ionized by more conventional ionization methods. So it is similar in character to electrospray ionization, that is ESI, that was mentioned in statement 1. So in that both techniques are relatively soft and it is low fragmentation basically. So these ways of obtaining ions of large molecules in the gas phase through molding typically produces four fewer like multi-charged ions. And this molding is a three-step process. The first is the sample is mixed with a suitable matrix material and applied to a metal plate. And the second step is a pulsed laser irradiates the sample, triggering ablation and desorption of the sample and matrix material. And finally, the analyte molecules are ionized by being protonated or deprotonated in the hot plum of ablated gases actually. And then they can be accelerated into whichever mass spectrometer is used to analyze them. Okay, so here the last statement is pending. So the last statement is small d analyte is mixed with aliphatic matrix and bombarded with matrix is bombarded, which fast beam argon or xenon atoms. This is called hard ionization. This statement is also incorrect. Okay, so this is also incorrect statement. Basically, in MALDI, the analyte is typically mixed with an aromatic matrix and ionized by a laser. So the use of aliphatic matrices are and bombarding with fast beams of atoms is not a common method in MALDI. So and this first statement is also correct, and the third statement is also correct. So the combinations of the options are 1 and 3 is the correct one according to this. The option B is the suitable one. So according to the theories and principle of the and all the statements, so the correct answer for the question 5 is the option B. That is statement first and statement 3 are correct. So moving to the next question, that is question 6. So question 6 states that a volunteer gave his skin cells and blood cells for whole genome sequencing, and he found that the genome sequence of both cells is exactly the same. So help him understand why the two cells look and function so differently if the genetic material is exactly the same. So the possible options here are option A is the two different cells mutate while development and happen to show exact same genetic sequence. And the option B is the proteome is static in all the cells. The option C is the genome of different cells is dynamic. The option D is the expression of proteins in various types of cells is different at a given time. So before coming to the conclusion of this question, let me introduce you what whole genome sequencing is. So, whole genome sequencing is also known as full genome sequencing, complete genome sequencing, or entire genome sequencing. So, in this process of this is the process of determining the entirety or nearly the entirety of the DNA sequence of an of any organism's genome at a single time. So, this entails sequencing all of the organism's chromosomal DNA as well as DNA contained in the mitochondria and for plants in the chloroplast also. So there are some things called alternative splicing. So alternative splicing is a cellular process in which exons from the same genes are joined into different combinations, leading to different but related mRNA transcripts. So these mRNAs can be translated to produce different proteins with distinct structures and function, all form a single gene. So where this means the exons are joined in different combinations leading to different mRNA strands. So consequently, the proteins translated from alternatively spliced mRNAs usually contain differences in their amino acid sequence and often in their biological functions. So here, let me explain some key points to help the volunteer as given in the question to understand why skin cells and blood cells, despite having the same genome sequences, like gene expression, some cell differentiation, and some tissue specific gene expression, epigenetic modification, some cellular function and morphology. So coming back to the question, so here, so according to that, the all the possible options, and that option D, which says that the expression of proteins in various type of cells is different at a given time. So according to this, the option D is the correct answer for the question 6, which states that the expression of proteins in various type of cells is different at a given time. So the correct answer for the question 6 is the option D. Okay, so moving to the next question 7. So question 7 says that in 2001, the first draft of the human genome was released. So the objective was to read the entire human genome from start to finish. Scientists from all across the globe confronted a number of challenges in sequencing the whole genome. So, which of the phenomena does not describe the issues that have arisen in the Human Genome Project? So, the given possible options here are so sequencing in multiple replication start sites, sequencing multi-gene sequence, especially in the shotgun approach, and the option C is sequencing the non-coding gene sites. And statement fourth and the option D and E are both A and B options are correct, and both A and C options are correct. So let me explain first that what this human genome project is first. Then we'll go back to the question and conclude that question. So the human genome project. So human genome project was an international scientific research project with the goal of determining the base pairs that make up human DNA and of identifying, mapping, and sequencing all of the gene and of the human genome from both a physical and a functional standpoint. And the other statement, the other term which covers in the question is called shotgun sequencing. Shotgun sequencing is a laboratory technique for determining the DNA sequence of an organism's genome. So the method involves randomly breaking up the genome into small DNA fragments that are sequenced individually. So a computer program looks for overlaps in the DNA sequences, using them to reassemble the fragments in their correct order to reconstitute the genome. Okay, so here assembly of DNA sequence reads an assembled DNA sequences, then DNA sequencing with genomic DNA, fragmented DNA, and DNA sequences reads. So in genetics, the shotgun sequencing is a method for sequencing random DNA strands. So in shotgun sequencing, DNA is broken DNA is broken up randomly into numerous small segments which are sequenced using the chain termination method to obtain their reads. So, multiple overlapping reads for the target DNA are obtained by performing several rounds of this fragmentation and sequencing. Then the last computer program then used the overlapping ends of different reads to assemble them into a continuous sequencing. So the conclusion is shotgun sequencing was one of the precursor technologies that was responsible for enabling whole genome sequencing. Okay, so now we'll look to the possible options for this. Here, the multiple replication start sites. So according to this, A and C sequencing and multiple replication start sites. So according to this, the statement, the option E is the correct one, which states the option A and option C are correct. That is sequencing the multiple replication start sites and sequencing the non-coding gene sites. Okay. So the correct answer for the question 7 is option E. So moving to the next question, that is 8. So here one condition is given below. So you are doing research on HPV associated cancers. So one of the main HPV oncoproteins is E6. So when the viral protein is expressed, human cells that have been infected with the virus turn cancerous. So you may learn more about the 3D structure and mutational pattern of the protein or gene with the use of potent bioinformatic tools and online servers. So please respond to the following inquiries regarding HPV-16 and E6 protein. And now the question is, what is the PDB ID of the C-terminal domain of HPV-16 E6 oncoprotein, and what was the biological method of acquiring the 3D structure? So this is basically a normal research, you can say a literature review. So all the information is given here, like we have given with the code of HPV-16 E6 oncoprotein. When we will copy this term in PDB ID on PDB website, we will get this kind of interface. So whenever you will put this HPV 16 HPV 16 and E6 oncoprotein, so this interface will come, which says that the 2LJZ is the structure of the C-terminal domain of HPV 16 E6 oncoprotein. And the next question what they were asking and is what was the biochemical method of acquiring the 3D structure. So here the method is solution NMR. Okay, so do you see anywhere this 2LJZ in the option? 2LJZ is in option 2, and as you can see solution NMR also was also there. So the correct answer for the question 8 is option B. This PDB ID is 2LJZ and method of acquiring 3D structure is solution NMR. So here the option B is the correct answer for the question 8. And one more thing students, and the question 9 and question 10 is also related to the same condition. So both the upcoming questions is related to the same thing. So coming to the next question, I told that the ninth and 10th question are correlating with the question 8. So here, what is the length of C-terminal domain of the protein? So please mention it in amino acids. Also write down the uniport ID of the protein it encodes. So as I told you, whenever you copy paste this HPV-16 E6 oncoprotein, in PDB ID will get this code called 2LJZ, and when you will put this in RCBD, then you will get this kind of interface which tells you it has the molecule of protein E6 and the sequence length of the amino acid is 75. Okay, and the organism is human papilloma virus 16. Okay, and the uniport, wait, in question 10 they are saying the uniport thing, just focus on this uniport portion also. So this will cover in the next question in 10. In question 10. So we'll still remain in the question 9. Hmm. So C-terminal domain of the protein of the mentioned in the question 8. So according to this, they have given this sequence length is 75 and the Uniport PDB ID is P03126. So according to this, the option A, the right answer for the question 9th is option A. Okay. And the next question is also related with the same thing. Wait, let me show you. So the question 10 says that what is the subcellular location of the above mentioned protein? Like above mentioned protein means in question 9 and question 8. So does it form any multimeric complexes? If yes, so please mention and pick the correct combination of the option. So the possible options given here are: it is a systolic protein, it forms homodimer in its active stage. So it is a mitochondrial protein, it forms heterodimer with E7 in its active stage. And the option C is it is a systolic protein, it forms homotetramer in its active stage. And the option D is it is a nuclear protein, it forms homodimer in its active stage. Okay. So when you will put this code, we are getting the PDB IDs P03126. And we will copy this PDB ID there. So we will get this kind of interface there. So which states that the subcellular location is the host nucleus and host cytoplasm. And the subunit is it forms homodimers, interacts with ubiquitin protein ligase with coding UBE3A or E6AP and thus forms a complex with human TP53 and it interacts with human NFX1 and MAG-I3. So coming back to the possible options for the same question as here, according to this, the option D is the right one. So it is a nuclear protein and it forms homodimer. It forms homodimer in its active stage. So the correct answer for the question 10 is option D. Okay, so this is for today. So you guys can
Excuse me, sir.
leave the session. So we'll meet in
Excuse
the
me,
next
sir.
session, which will be on next Tuesday from 7 to 9.
Sir.
So thanks for the day.
Hello. Excuse me.
Oh, hello. Hi, Vanshika.
Hi, sir.
Sorry, my mic
Am I
was
audible?
my speaker was on mute. Yeah, yes, you can say.
Okay. Sir, can you please go back to first question once?
Question one.
Yes, sir.
Okay, wait. Hanji.
Sir, I just wanted to know why
Hmm.
the fourth option is not a correct option. Like
That
in an
aquaporin.
explanation,
That
like
aquaporin
in
one.
yeah, aquaporin is not considered as quaternary structure, but hemoglobin is. Isn't hemoglobin quaternary?
See, as I told you, here. See, the hemoglobin is a classic example of a protein with quaternary structure, and
Yeah.
hemoglobin is composed of four subunits. Okay, and each containing a heme group for oxygen binding. That's why it is called a with this quaternary protein structure. And in other hand, the aquaporin, which is called as the water channels, and are channel proteins from a larger family of some, and it does not contain that four subunits. That's why aquaporin is not considered as a quaternary structure, but hemoglobin is, because hemoglobin have that four subunits, each containing a heme group and of four oxygen binding sites. That oxygen binding sites are not there in that aquaporin structure. Am I clear?
Yes, sir. But
It is a
now,
tetramer. It is a tetramer. Like it forms four part of clusters, but it is not a quaternary structure of a protein. The tetramer and quaternary structure both are different here.
okay, okay, sir. All right.
Hanji.
Thank you so much.
Okay, Vanshika. So we'll you guys can leave the session. We'll meet in the next Tuesday. Okay, thanks for the day.
Thank you, sir.
Thank you. You're welcome.